Hello, my name is Sергєєва Жанна, and we are talking about plasmids. This is actually our second meeting. In our first one, we discussed basic things about plasmids, the definition, what are plasmids, and just some facts about their copy number, their size. And today we continue our conversation about plasmids, their diversity and necessity for bacterial cells. So let's go on. Well, today I would like to tell you about incompatibility of plasmids, because not all the plasmids can coexist in one bacterial cell. What does it mean? It means that some plasmids cannot live together in one bacterial cell because they are too similar in some their qualities or properties. Usually we talk about their replication abilities. It means that they have a very similar replication, a lot of similarities in their replication mechanism, and they cannot replicate together. It means that they cannot maintain the cells in one bacterial cell when they are present together, and they are incompatible. They cannot coexist. If plasmids are present in the cell, different types of plasmids, and they can coexist, it means that they are compatible. If they cannot, they are incompatible. Of course, a good question is how can two incompatible plasmids meet in one cell? If they cannot coexist, how they can get inside the cell? What's the meaning? We're going to return to this question a little bit later, but now we're going to understand what exactly is incompatibility. So, incompatibility, as I already said, two plasmids cannot coexist together. Different types of plasmids can exist together in one cell if they belong to different incompatibility groups. How can plasmid leave the cell if it's incompatible with the other plasmid? Well, first of all, how plasmids distribute through cells. If we have a cell and it contains a plasmid, and it starts the division and replication, it means that when the division occurs, each daughter cell receives a copy of a plasmid. Sometimes it happens due to the high copy number of these plasmids. It's like there are present several hundreds of these plasmids in the cells, and when it divides, simply each daughter cell contains their own number of copies. Or if there is a megaplasmid and only one copy number, so it just replicates with the chromosome, and when the cell divides, each daughter cell receives its plasmid, and we have all the further generations containing these plasmid. So the plasmid inside the cell, then everything's okay. If we have cell cycle with incompatible plasmids, it means that they cannot replicate. They are too similar in their replication mechanism, and when the cell divides, each daughter cell receives its own plasmid. Not a copy of it, but its own type of plasmid. One goes to one daughter cell, and another goes to another daughter cell. And actually, on the genetic level, we have two different strains. So we had one strain with two plasmids, and now we have two different strains with only one plasmid in each of them. There can be also phenotypical differences, and we are also going to return to this a little bit later. To be able to use plasmids as vectors, it is also important to have the information about their incompatibility with other plasmids. And that's why there were created and systemized some incompatibility groups. Well, there are more than 20, almost 30 incompatibility groups right to this moment. They have their names with Latin letters, and if plasmids belong to one incompatibility group, they cannot coexist together. If they belong to different groups, they can exist together in one cell. For example, PUC plasmid of University of California. It's a very famous vector, small plasmid. It is presented in 5 to 7 copy numbers per cell. It means that there are up to 700 copies of this plasmid in one bacterial cell. And it belongs to incompatibility group A. If we talk about another vector, PBR 322, it's also a very famous vector. Copy number is much lower. As you can see, it's only 15 or to 20 copies. And those two vectors cannot coexist together in one cell. We cannot put them together in one cell. If our cell already contains one of these vectors, we cannot put another one to this cell. But we can put a vector from another incompatibility group, like PR6K. We can use it if our cell already contains any of the vectors from another incompatibility group. So that's the idea of understanding incompatibility, to be able to use, for example, like different plasmids in one cell, and to understand why one plasmid can eliminate and another stays within the cell. So a good question is, how can two for example, plasmids from one incompatibility group appear in one single cell? If they cannot coexist, how can we actually talk about them meeting together? It happens due to the vertical and horizontal gene transfer. Well, before we start to talk about horizontal gene transfer, we have to talk about vertical. It means like from mother cell to daughter cell. So it's the easiest way for plasmid distribution, for genes distribution between cells. The cell divides and each daughter cell receives its copy of a plasmid. Another variant is when a plasmid integrates into the chromosome. It happens rarely, but anyway, it happens in nature. Usually, plasmids are autonomous. They are out of the chromosome, but sometimes they can integrate into the chromosome. If you are familiar with these facts about bacteriophages, you must know about prophages. Well, some bacteriophages, after entering the cell, they integrate into the genome. They integrate their own genome into the bacterial chromosome, and from this moment we start calling them prophages. And when the cell divides, its chromosome replicates, and each daughter cell receives a copy of a chromosome with the integrated prophage. The same happens with the integrated plasmid. The chromosome with the integrated plasmid replicates, and each daughter cell receives its own copy. Anyway, it receives it for sure. So, this is a vertical gene transfer from parents to children. All the children have their own copy of a plasmid. But there is also horizontal gene transfer. Well, we can say between the cells of one generation. It's like we're not talking about new generations receiving their genetic information from parents, but we're talking about the spread of genetic information between already existing bacterial cells. So some bacteria in nature can take the genetic information from the outer space, from the environment. These cells must be in the state that are called competency. So these cells must be competent, a special state in which the cell can take in the genetic information and probably use it. So if we talk about plasmid, it can get inside the cell. There are also some other mechanisms. We are going to return to them, but now we're talking about the transformation, because this process of getting the DNA from the outer environment is transformation. These cells can transform. We can use this mechanism in the laboratory, and we can make the transformation, perform the transformation even to those cells that are not able to do it in nature. We can perform special procedures to make almost any bacterial cell competent so that it can take in the information from the outside. So one of the possible ways is chemical transformation. It is presented in a very low temperature, on ice we call it. So the cells, they freeze, and we use some chemicals to make them even freeze more. And what happens with the cell? Its membrane becomes very rigid. So usually membrane is flexible because it contains lipids, and it becomes very rigid, and we put the DNA into the tube and it adjusts to the bacterial cell, and then we perform a heat shock. 42 degrees, it's almost the border temperature, because the high temperature will simply destroy proteins, but 42 degrees, it's a heat shock, but it doesn't destroy anything yet. So, and this heat shock makes the cell, you know, like in a moment take in any DNA molecules that were on the surface of the cell. So it takes in those molecules, and then we again put the cells on ice so that the membrane will kind of like close these pores through which the DNA came into the cell. And this is called chemical transformation. Easier way is to perform electroporation. We use electricity on cells, on bacterial cells, to create pores in the membrane so that the DNA will get inside. And it's a very easy procedure, usually performed when we talk about vectors. So you can see it's not simply a plasmid, it's a vector that contains different genes. What's wrong with the video? It's a vector that contains genes of interest. It's a factory, little factory, and after the influence of electricity, we put it inside the cell, and we have this cell transformed with the vector, with the plasmid. So, and this is the way how we can put a vector inside of the cell. So transformation, horizontal gene transfer, is an approach that can make it for two different plasmids to make it possible for two different plasmids to meet inside one bacterial cell and to discover whether they can coexist or one of them has to leave. So this is incompatibility of plasmids. And transformation, it means that if our bacteria meets some sort of new information and it decides to take it in, this bacteria must be ready for phenotypical changes, because these genes must do something new, can do something new to our bacteria. If we talk about biotechnology, so these are genes of interest and we change these bacteria as we want it to. But if we talk about nature, so something happens with our bacteria when it takes in new information. So what information can actually carry those different plasmids? First of all, there are conjugative plasmids. One of the most famous is F factor, fertility factor. Well, these plasmids make it possible for bacterial cells to perform conjugation, a process of exchange of genetic information. Due to this conjugation, if we have a cell that contains F plasmid and another one that has no F plasmid, so there creates a conjugational bridge between those cells, and one copy of the plasmid travels through the bridge to another bacterial cell. And this is conjugative process, and some plasmids code this ability for conjugation. There also are plasmids, resistant plasmids, that code resistance to antibiotics and some other substances. But resistance to antibiotics is the most important one, because due to these plasmids, genes of antibiotic resistance spread through different species of bacteria, and they cause the problem that exists in the world today, the resistance of bacteria to antibiotics and our inability to cure some diseases. So some of the plasmids are really responsible for this fact, like RP4 plasmid. Also, well, it was widely used as genetic instrument. So there are also coplasmids. These plasmids code cholecines, structures that are like toxins. They help one bacterial cells kill another bacterial cells in the competition of survival. Virulence plasmids. Also, these plasmids, like our plasmids, are really very important to understand because these virulence plasmids, they can make a harmless bacteria a very dangerous pathogen. Because if we have, for example, a Escherichia coli that is absolutely like harmless, and it floats somewhere in the water, and it meets these virulence plasmids and takes it in, and it becomes very virulent, and it produces endotoxins, and it can cause the disease of human, and it becomes really dangerous because of this virulence plasmid. So also one of the most famous plasmids is TI plasmid of Agrobacterium tumefaciens. Agrobacterium tumefaciens is a phytopathogenic bacteria. It means that it causes the disease of plants. But anyway, why this TI plasmid is very famous? Because it causes tumor inductions. The name of the plasmid, TI, means tumor induction. It causes tumor induction in plants. It means that this plasmid can travel through kingdoms, from the kingdom of bacteria to the kingdom of plants. It travels from the cell of bacteria to the cell of plants and causes tumor induction. And in a very wide range of plants actually suffer from these pathogens, suffer from the influence of this plasmid. And this plasmid is widely studied because of this particular property. Metabolic plasmids, they give different bacteria an ability to use different substances in their metabolic processes, and it means that it's a new pathway to get energy and material, so it's also like a privilege in the competition of survival. And I think that this will be enough for today, and we are going to continue our conversation about plasmids and focus on the creation of vectors, isolation of plasmids, and actually how can we use these structures in biotechnology. Now, thank you for your attention and goodbye.